Introduction to pharmaceutical microbiology. So, microbiology is the science study of microorganisms. It is the study of microscopic organisms that are too small to be identified with ordinary eyes. So, these organisms could include organisms like algae, we have bacteria, we have the virus, we have algae, we have the fungi, we have the protozoa. So microbiology is an experimental biological science that deals with invisible forms of organisms. The study of microbiology is often synonymous with study of bacteria, but apart from bacteria, microbiology also studies virology, mycology, algology, protozoology. Microorganisms are agreed to be organisms less than 1 millimeter in diameter, except fungi and mold. These are the applications. We have agriculture, we have pharmacology, we have biochemistry, etc. So these are the application of microbiology. So microscope is the optical instrument which can magnify small objects which cannot be seen by naked eye, which are the microorganisms. So they were invented in the 17th century, and they include two types depending on the lenses, which are simple microscope and a compound microscope. The characteristics of microorganisms include small sizes, no cellular differentiation, and they are present everywhere. So these are the characteristics of microorganisms. You can pause the video to learn about it more. Let's talk about some special microorganisms and their specifications. We have the protozoa, which are eukaryotic, have cilia, flagella, pseudopodia for movement. They are animal-like in that they ingest particulate food, and they lack rigid cell wall and do not contain chlorophyll. The study is protozoology. They are differentiated on the basis of morphological, nutritional, and physiological characteristics. Their role in nature is varied, but the best known protozoa are few that causes diseases in human beings, such as the Toxoplasma gondii, which causes toxoplasmosis. Other examples include the Plasmodium species that causes malaria, and Trypanosome species that causes sleeping sickness. So we have the algae, which varies from 1 micrometer to several feet. They are considered plant-like because they contain the green pigment chlorophyll, which carries out photosynthesis. They are either motile or non-motile, that is for the algae. So yeah, you can pause the video. They causes problem by clogging water pipes and releasing toxic chemical into water bodies or growing in swimming pools. Then the next one is the fungi, which are saprophytes or parasites. They also have eukaryotic cell structures like algae, and they have rigid cell walls. So that is about the fungi. They form fruiting structures called conida or exospores and endospores. They are present in air, dust, and soil. Their size ranges from 2 to 10 micrometer. That is why they have larger sizes and can be referred to as microorganisms. So these are some of the applications. Their usefulness. Then we have the bacteria as well. They're ranging from 1 to 5 micrometer and have rod, coccus, and spiral shapes. They are actually prokaryotic, not eukaryotic. And cell division is by binary fission. We also have some of their roles. So this is about the actinomycetes, which are vegetative bacteria and are non-motile. They have fungal morphology. Can pause the video as well. Then we have the virus, which are ultra microscopic and then non-cellular obligates parasites of plants, animal, and bacteria, as well as other protists. They can only be seen on an electron microscope, unlike cells. They have only one type of nucleic acid, which is DNA or RNA. The study is known as virology, and it causes a whole lot of diseases, as you can see on the screen. Now let's talk about the discovery of microorganisms. So microorganisms were mostly discovered by amateurs. Example is Anthony van Leeuwenhoek, which is the father of microbiology. So we constructed many microscopes with single lens, which consisted of a spherical lens mounted between two metal sheets or silver or brass. So we have a spontaneous generation, which explains the origin of the course microbiology. You can pause your screen to take a look at it, as I'm trying to shorten the time of the video. So after that we have the transition period, which is theory of abiogenesis, which disapproved abiogenesis. This is the theory of biogenesis, which disapproved abiogenesis theory. So here are some of the scientists that contributed in a way or two in trying to disapprove the theory of abiogenesis. So take your time. can pause the screen and take a look at the scientists that tried in a way or two to disprove the theory of spontaneous generation, which basically means that it basically stated that microorganisms were generated from non-living constituents. So these are the processes that was carried out to constitute what is today known as the theory of biogenesis. Then after that, we have the classical Rodin era, which is the germ theory. Yeah, this is a transition from the transition theory. Yeah. So we have two key major players in the classical Rodin era, which are Louis Pasteur and Robert Koch. So you can pause your screen to see their major contribution. So basically both the classical Rodin era and the biogenesis theory were all trying to disapprove the abiogenesis theory of spontaneous generation. They are all trying to come up with reasons, facts, evidences of why living organisms never came from non-living constituents. So that's just basically it. So these are the awards won by some of the contributors, and here is rule from Robert Koch. So that is it. Remember to pause your screen to understand more. So we are going to see the contributions. Such as isolation of viable cholera. These are contributions from Robert Koch actually. So he isolated viable cholera, which is the cause of cholera, and other contributions as well. And then we have the modern molecular biology. This is basically the era we are now. So here are some contributions in molecular biology, which is basically the area we are right now. You see some of the contribution of some of the scientists in molecular biology. So that is basically it. So that is it.